As dear children, the presence of starch in this leaf should be proved. So which chemical we need? We need a chemical called iodine or betadine. So the chemical used or the reagent used to test the presence of starch is nothing but called iodine. Here, what reactions is shown by iodine? If starch is present in this, if you add iodine on this leaf, it will turns into bluish black color. Bluish black color. If anything turns into bluish black color by the addition of iodine, then the material is said to be a starch. So iodine is a great reagent to prove starch is present on anything or not. If you add iodine drops, if the material turns into bluish black, that is called a positive result of iodine, tells that starch is present. If the iodine gives a negative result, the material doesn't turns to bluish black. If the material doesn't turns to bluish black, means starch is not there inside. Here we need to know one more thing, one more barrier is there. What is one more barrier? We are simply telling that iodine is used to test the presence of starch. If you add the iodine drops on this starch, on this leaf, if it turns into bluish black, we can directly tell that yes, starch is present. But iodine doesn't react directly on leaf. Here is an exception. A barrier is there in this experiment. Why iodine directly doesn't react on leaf? Because leaf has a different pigments. Is it or not? We need to know about pigment here. I am telling you that iodine can test the presence of starch in leaf. Yes, starch is present in leaf, but iodine cannot directly react with the starch inside leaf because there is a barrier in the middle. There is obstruction in the middle which is called pigment. So what is pigment means? Pigment are coloring agents, coloring substances in a living structures. Skin has a pigment, is it or not? Called melanin. Leaf has a pigment called chlorophyll. So pigments are color imparting chemicals in different living structures. Blood has a color called red color because of a pigment called hemoglobin. So the different living structures with different parts are with different colors because of pigments. So pigments are color giving chemicals in living structures. Understand? Like that leaf also has a different pigments. Mainly it has a pigment called chlorophyll. That's why this leaf is in green color. So this chlorophyll will obstruct, will interfere in the middle, will act as a barrier in the middle not to react iodine on this starch. So what to do now? We have to remove this pigment called chlorophyll. First, if you remove the chlorophyll from the leaf, then iodine can react directly on the starch, giving a positive result called bluish black color. So how to remove this chlorophyll now? We want to remove chlorophyll, the middle interfere agent called chlorophyll, to test iodine to prove starch is there or not. To remove chlorophyll, we need one more chemical. We need two main chemicals in this experiment. One first chemical is iodine to test the presence of starch, but iodine cannot test it directly because a middle barrier interferer is there, that is called a chlorophyll pigment. Now if we remove this, iodine can be tested directly. The starch is present or not. To remove this chlorophyll, we need to use one more chemical called methylated spirit. So we need to take one more chemical called methylated spirit, alcohol. Why should take methylated spirit in this experiment? It is used to remove chlorophyll. Then the leaf will become chlorophyll less leaf without chlorophyll. Then iodine can be tested, used to test the presence of starch. In what way we have to use this methylated spirit to remove chlorophyll in the leaf? We have to boil the leaf in methylated spirit. So methylated leaf cannot remove the chlorophyll directly. If you boil the leaf in a methylated spirit, then the pigments are slowly removed and the leaf will become pale white in color. Pale means colorless. Where the colors are lost, that is called as a pale color. Now we need to boil. So boiled methylated spirit must be prepared to keep the leaf inside, to remove the pigments. Then if you test with iodine, the starch is proved with a positive result called bluish black color. Now set up the arrangements to do all these things. First to boil the methylated spirit and then adding iodine to remove, to test the presence of starch. And one more important difficulty here, to boil the methylated spirit is a difficult thing, that methylated spirit is a flammable substance. Flammable means it can catch fire easily, just like a petrol. So how to boil it? So we will set up an arrangement to boil the methylated spirit without touching to fire. So let us see the arrangement here. Yes, dear children, we arranged these apparatus in such a way as shown in the textbook. How we arrange? Let us see the apparatus. First take a beaker. Take a beaker with a level of water in it as we taken inside. Take a beaker with a level of water and then take a test tube. Take a test tube with which is filled with methylated spirit. So different apparatus with different arrangements I am showing, and they are all combined to make this such an arrangement. First we have taken a beaker with water and a test tube with methylated spirit. Methylated spirit. Understand to you. Later take the leaf to be tested for the presence of starch. Take the leaf to be tested for the presence of starch which is exposed to sunlight into this test tube. So we have taken the leaf into test tube containing methylated. Dip it into the test tube. Dip the leaf into the test tube with the methylated spirit. Now keep this arrangement of test tube with methylated spirit and the leaf to be tested inside the beaker containing water. So beaker with water, test tube is with methylated spirit and it is with the leaf to be tested. It is there already here. Now we need to boil, heat the methylated spirit. Why to heat methylated spirit? If you heat and boil the methylated spirit, then the leaf will lose its pigments. Then only it is allowed to test with the iodine for proving starch is present or not. So here one important thing is there. We should not boil the methylated spirit directly. That's why we kept this test tube with methylated spirit in water containing beaker. Now we arranged a heat source. Now this heat source called Bunsen burner will heat the water, will boil the water. With that heat the methylated spirit get boiled. So we are boiling methylated spirit without touching to the fire directly. So a boiled water bath is prepared here. So water is boiling, by the heat the methylated spirit is boiled. So here is an important question, dear children. Why test tube with methylated spirit is not directly faced to heat source to boil it? If it is boiled directly on this heat source, it may catch fire and the leaf will burn and the experiment will fail. So not to happen so. So this is a first important question here. Why methylated spirit is not boiled directly on a heat source to remove pigments? It is an flammable substance. So it should be heated in the water, then the methylated spirit will be boiled. We will continue.